All right, sorry, minor technical difficulty. Okay, we all ready? Have notes out, ready to go? The first couple slides here focus mostly on, hang on, it's not presenting to the people at home, so I fix that so they can see. There we go. Let's see if we're all set now. Better. Okay, now we're ready. The first few slides today feel a little bit disjointed, like they're kind of separate topics, but basically the point of these couple slides stuck in here is to remind you that genetics is really complicated. When we learn about genetics, we tend to learn about traits that are very clear in terms of how they're inherited. They're dominant or they're recessive. Even things like codominance and incomplete dominance, it's either this or that or the other thing. Whatever genes you have tells you exactly what the phenotype will be, and it's pretty clear. But in real life, of course, there are a lot of characteristics that are influenced by a lot of other factors. So pleiotropy is the first example of how this can be more complicated to figure out. Sometimes one gene, so we're still talking about one pair of alleles, you get one from mom, one from dad, but it can affect multiple parts of your phenotype. So it may not be as simple as saying this gene gives you a widow's peak or not. Sometimes one gene can impact several different things, and that's kind of what this is showing. And there are a couple of examples there. There's a gene in fruit flies. Drosophila is the genus of fruit flies. It's one you'll hear a lot if you do a lot of biology. It makes their eyes white, but it also tends to make their flight muscles defective. That's an effect of that gene. It does both of those things. Albinism in humans, if someone is homozygous recessive for that gene, they don't have pigment in their body, but it can also affect their eyesight and their hearing as well. So it has multiple effects. You can also think of this, some genes code for, trigger the production of hormones that then travel throughout your body and have lots of different effects depending on the types of cells that receive those signals. So it's not always as easy as saying one gene causes one specific characteristic. Sometimes one gene can cause several different things to happen. Sometimes it's also not just genes. With these terms, I'd really suggest that instead of just trying to memorize definitions, you really look at what these words mean. If we break this one apart, what does the prefix multi mean? Many or lots. And what is factorial? What does that sound like? Factors. Many factors. So it's not just the genes that determine the phenotype in some cases. So multifactorial inheritance means many factors influence a phenotype. It's partly genetically determined, and you can probably think of examples of this in humans. Things like your cholesterol level. Part of that is genetic. Part of that is how your body processes things and how it clears out cholesterol. But part of it is also your diet and how you live your life, right? It's a combination of things. So you can think of this with a lot of things. Here's another kind of example. Siamese cats have a gene that codes for darker pigments. That gene that makes that darker pigment tends to be more active at lower temperatures. And you can actually see that when you look at this cat. Turn and explain to the person next to you how you can see this in its phenotype. Turn and explain that to each other. Thank you. So I heard most of you say this. The parts of the cat that are colder are the ones that are darker, right? Ears that stick out, nose that sticks out, paws that stick out, the tail, the core of their body, that tends to be the warmest part of you. That's true of humans too, right? Your fingers and toes tend to start getting cold first, and your core stays warm. And so because the extremities, the paws, the nose, the ears get cooler, they tend to express more of that gene and produce more of that darker pigment. I don't know specifically about this exact example, because it probably depends whether that's something that's kind of determined at a certain developmental stage, like that's when the pigment is produced, and then it just can be either that gene is on or off for the rest of their life, or if it's something that's continuously changing. I don't know that. It's a good question, though. And there are lots of traits like this. There are some flowers where the color of the flower is genetically determined, but can also be influenced by like the pH of the soil or things like that. Those are other examples of multifactorial inheritance. Here's another one where the word really tells you what it means. What does poly mean? Many. What is genic? What does that refer to? Genes. So polygenic inheritance means there are multiple genes that determine one trait. Polygenic inheritance is really common anytime you think of a characteristic that varies along a continuum within a population. So if you think about something like human height, height is both polygenic and multifactorial, because obviously some things like the quality of your healthcare and your diet and the cleanness of the air you breathe and the water you drink, all that can affect your health and how tall you end up being. But some of it's also genetic, so it's multifactorial, but it's also polygenic. There are a lot of genes. It's not just you're tall or you're short. People can be a whole wide range of heights, and that is impacted by many, many genes. The other place this is usually true is anything that has to do with color. If there's more than just two choices, right? If you're talking about like birds that are either blue or red, that's a different thing. But if you think about like eye color, there's a whole wide range. Or height, I already talked about that one. Or skin color, where there's a whole range of colors. Hair color is probably polygenic as well, because there's a whole range. This is kind of a simplified version, but it gives you an idea of how this might work and how you can think of polygenic inheritance. This table shows 3 genes that code for hair color or skin color or something that happens on a continuum from light to dark. The genes are A, B, and C. And basically, you can think of each dominant allele as like one, I don't know, pick a unit, one unit of color. We'll just say that. So someone who gets 3 recessive alleles from mom at the top and 3 recessive alleles from dad at the bottom has 0 dominant alleles down here in the bottom right, and they are going to have the lightest color skin or hair or whatever this is. And then the more, so like somebody right here would get 1 dominant from one parent and 1 dominant from this parent, they have 2 dominant alleles, and so they get a little darker color. And so the more dominance, like I said, this is a simplified way to think about this, but it kind of shows you how a whole bunch of genes can kind of interact with each other to produce a range of different colors or heights or whatever. So anytime there's a trait that doesn't just have a couple distinct choices, it's probably a polygenic trait where lots of genes interact to determine those characteristics. Questions about any of those? All right, so let's go back to a little bit of simpler genetics. Obviously, one of the major reasons that people spend a lot of time studying genetics is because they want to be able to trace inheritance of specific traits, often disease-causing genes, within families. I don't think I printed this one for you because there's not really any information. There's just a comic about Piglet and what's this dude's name? Porky Pig, is that who that is? The guy who stutters a lot. But being able to look at relationships and trace genes through generations can be really important if you're trying to determine your risk of getting a genetic disease or passing a genetic disease on to your children. So that's the main use of a pedigree. This one you probably have printed, right? Did I print that? I thought I skipped that one, so I took it out. Nope, we didn't talk about I forgot I left the epistasis one in there. I thought I took that out this year. Epistasis is another example of complexity, and I'll just talk about it because you have it printed in your handouts. It's one that I don't think you have to know for the AP exam. I think it's kind of a cool example because a lot of people have labs, and that's kind of the classic example of epistasis. Epistasis literally means something like one gene on top of another. So like I said, a really good example is coat color in labs. You can have chocolate labs and black labs and yellow labs, right? There are two genes that determine coat color in labs. One of them determines whether the pigment is produced. The other determines whether the pigment is actually deposited in the hair so that you can see it. And I'm blanking right now on exactly how it works. To be a yellow lab, they have to be recessive in both of those. They don't have, well, no. Take that back. I'm gonna have to look this up now that I deleted it.
So
does it explain it on there? Let me read.
It just says like no pigment deposited.
Oh, there it is. Okay. So it explains it to you in here. The dominant color is black, the recessive color is brown, but the second gene determines if it's deposited in their hair. So whatever their genotype for the color is, if they have black or brown fur, if they have at least one dominant allele for the second gene, then that color shows up in their fur. But if they're recessive for the second gene, it doesn't really matter whether they have black or brown pigment being produced because that pigment isn't put into the hair where you can see it.
So labs can be black or brown, but still be like they have the black pigment being produced, but it's still the red.
Yes. That is true. So I would assume, I don't know if anybody's ever tried this, I would assume that if you took a yellow lab and somehow were able to turn on or to give them a dominant copy of that gene so they would start depositing pigment, maybe you could change their color. I don't know. So yes, epistasis you don't need to know, which is why I took it out of this one, and I thought I had skipped printing that one, but apparently I didn't. All right. So now we'll move on back to pedigrees. Hopefully pedigrees are something that you are fairly familiar with. It's basically a genetic family tree, right? So this first slide just goes over the basic symbols. The squares are males, the circles are females. Sometimes you will see people with a slash through them. Those are deceased members of the family because a lot of time if you're making a pedigree, especially to figure out inheritance of a disease or something, even if someone's no longer living, it's important to know whether they ever showed any signs of that disease because that can help you figure out how it has moved through generations of the family. So pedigrees will often include deceased previous generations if you have information about whether or not they showed that trait. Couple important things to remember. Just because a trait is shaded, it actually doesn't tell you anything other than that person showed that trait. Unless there's information written around the pedigree that tells you this is a dominant disease or this is a recessive condition or whatever, a lot of times when people start building pedigrees, they don't know how a trait is inherited. They just know whether someone has a disease or not. So you shade in people who show the trait you're interested in, and then sometimes you can look at a pedigree and figure it out. But if all you see is a pedigree like the one on the screen, this could be a dominant or a recessive trait. It could be an autosomal trait or a sex-linked trait, which we'll talk about later. So it's just important to remember they're not always dominant, they're not always recessive. It just means that person has the trait. The other thing is that sometimes on a pedigree you'll see people who are half shaded, which means they are carriers. The word carrier means they carry the gene but do not show the trait. So what does the genotype of a carrier have to be? Dominant and recessive, they have to be heterozygous. So anytime you see the word carrier in genetics, that automatically means they're heterozygous for that trait, because it means they carry the allele for whatever disease or condition that you're talking about, but they don't show the trait. They have a dominant gene that covers it up. We can only really use the term carrier with a recessive condition. You can't carry a dominant allele without showing it, so that term just doesn't really work there. So pedigrees can give us a lot of information about characteristics. Like I said, one thing we can do with a pedigree is figure out whether traits are dominant or recessive. And sometimes you can also determine whether or not they're sex-linked, but we're not going to worry about that right now. This is a pedigree for widow's peak. So that means since it's a pedigree for this trait, the people who are shaded have a widow's peak, which is when your hairline in front comes down to a point in the middle of your forehead instead of going straight across. That point's called a widow's peak. So study this for a minute. Turn and talk to the people around you. Is widow's peak a dominant or a recessive trait? And where in the pedigree can you figure that out for sure? Ashley and Kylie, I can't mute my computer right now because I'm recording, but you guys can look at it and see if you can figure it out. And if you want, you can type in the comments and kind of chat about it that way. Is widow's peak dominant or recessive? How do you know?
Because with Joseph and Alicia, they don't have it, and now one of their kids does it, so Joseph and Alicia have it.
So here's my biggest hint with pedigrees. If you can, look for a family that has this pattern, that both the parents are the same and they have kids that are different. And it actually works both ways. You'll see in the next one, I think, an example of the other way. But in this case, both Joseph and Alicia have this trait. One of their kids has it, but the other one does not. So if you're not sure about inheritance, you can always guess and just try it out and see if it works. So if we use W's for a widow's peak, if it was recessive, which it's not, but this is how you know it can't be. If it was recessive, both of these people and this person, they all have to be homozygous recessive, right? It's the only way to show a recessive trait. Angela would have to have at least one dominant allele. Doesn't matter what the other one is. Is there anywhere that Angelo could get a capital W from these parents? No. So this cannot be a recessive trait. So this has to be dominant, because then what are the genotypes of the parents? Yeah. They have to be, if Angelo, so when you're looking at a pedigree, once you figure out whether it's dominant or recessive, the people who show the recessive trait, you can always determine their genotypes right away because they have to be homozygous recessive. In this case, once we know widow's peak is dominant, we know each of these people has to have at least one capital letter. Then you kind of look a generation up and a generation down to try and fill in the rest. So in this case, Angelo had to get each of those two recessive alleles, one from Joseph and one from Alicia. So each of them had to be heterozygous. In this case, can we determine for sure what Janice's second allele is? No. She could have gotten two dominant alleles. She could have gotten a dominant and a recessive. So sometimes you can't tell for sure. That's okay. But we do know for sure that this is a dominant trait, because otherwise these relationships do not work. All right, here's one more. The shaded people in this family cannot taste the chemical PTC. Have you guys ever done this taste test before? You get a little strip of paper and you put it on your tongue, and some people taste something super bitter and other people taste paper. Have you ever done that before? You might have. I can see if we have any of this paper, and maybe I'll bring it up tomorrow after the quiz or something. It'll be really fun. But in this case, the shaded people cannot taste this chemical The inability to taste, so that's what you're looking at. The shaded people here, in this case, is that dominant or recessive? Turn and talk to each other. Once again, look for the pattern where the two parents are the same, but they have a kid who's different. So this time, the family that helps you figure this out is this one. Because in this case, both the parents can taste PTC, but they have a daughter who can't. So what does the shaded trait have to be? What's the only way that that's possible? Yeah, if this, the shaded trait down here, is recessive, then just like last time, both parents could be heterozygous, and she could have inherited the two recessive alleles. It doesn't work the other way. Does that make sense? So in this case, the inability to taste must be recessive, because that's what was shaded here. Any questions about that? I think I put it on that previous slide. There is one more practice set of problems that's attached in Classroom today that's got a whole bunch of simple pedigrees, and for each one it asks you how that trait could be inherited. You can't always determine it for certain, but you can often eliminate a couple modes of inheritance. So if you want a little more practice with pedigrees and trying to figure out the genotypes of the family and all that kind of stuff, you can try some of those. Now we're going to move on. We're going to go back to just a little bit of history, and then we're going to talk a little bit about chromosomes. So in your textbook, we are moving here into chapter 12. Up to this point, we've mainly been focusing on individual genes, because remember, Gregor Mendel did that work with pea plants and talked about these factors that are passed from parents to offspring and called them genes. And he figured out that only one copy was passed on and that they sort independently so they don't affect each other. But he did not know chemically at a molecular level what he was talking about. So it wasn't until some years later that people kind of put together Mendel's observations with some other research to come up with what's called the chromosome theory of inheritance. And here you don't need to know these dates, but this is just kind of to put all this stuff together so you can kind of see how scientists figured this out. Mendel was doing his work in about 1860. Obviously, he did it over a period of several years. But it wasn't until 1890, so a few decades later, that the process of meiosis was seen and described. And the reason meiosis is important is that you're actually seeing chromosomes moving in cells and moving into egg cells and sperm cells and into new generations of creatures. And it took a few years even after that for people to kind of put that together, to say, hey, Mendel talked about these things that are passed from parents to offspring that separates, only one of them goes to the kids. And then we looked through these early microscopes and we saw these things moving around and moving to the next generation and separating, so only one is passed on to kids. I bet Mendel's hereditary factors are located on chromosomes. And so that's where they started kind of putting this together to figure out that Mendelian genes, what he had talked about it as his hereditary factors, have specific loci. The singular of this word, because you might see it a lot, is a locus. It means a position. It's the same root word as location. So if you think of the word location when you see those words, that might help you remember. So Mendelian genes have specific loci or locations along a chromosome. I do not know exactly what year he died. He obviously could have been. He wasn't super old when he did his work with pea plants or anything. So he definitely could have been. I don't know. You'd have to look up what year he passed away to see if he was still doing his stuff then. So then they were able to kind of take Mendel's laws that he had come up with, the fact that the two copies separate and only one goes to the offspring, and that different genes don't affect each other, and apply those to this idea of chromosomes and realize if the genes are located on the chromosomes, it's the chromosomes that are segregating. So only one of each chromosome goes to an egg cell or sperm cell. And the chromosomes and the genes on them that are sorting independently, meaning whichever copy of a chromosome 1 goes in this cell and that cell doesn't have any effect on chromosome 14, right? They're each just kind of doing their own thing. So it kind of took Mendel's ideas to the next level and started filling in some of the molecular stuff that he didn't know. Need to worry about that. So here are two pictures of human chromosomes. Take a minute and just turn and make some observations. Describe what you're looking at and talk about what you know about these chromosomes. You can also compare the two. These are called karyotypes when you have all the chromosomes laid out like that. You can compare and contrast the two and talk about what's similar and what's different. So turn and talk about those just for a second. All right, what kinds of things did you notice up here? So one important thing to remember when you're looking at actual karyotypes, these are actual photographs of actual chromosomes, which are not always nice, perfectly straight lines the way that we often draw them in pictures and textbooks. They are kind of floppy, and so they don't always lay nice and straight. When scientists make a karyotype, generally what they're doing in this type of test, this is an actual genetic test that they can do for people, is they take all the chromosomes out of a cell and lay them out in pairs. And normally what they're doing is they're doing a count to see if you have the correct number of chromosomes in your cells. And so sometimes they try and line them up as nicely as they can because that's how they figure out which pairs match, but they don't always lay nice and straight. What else do you notice?
That one doesn't have a Y chromosome.
All right, so the last pair down here, obviously the first 22 pairs in both, even though they're not perfectly straight in both, other than that, they're pretty similar, right? Chromosome 1 is really long. It has a similar banding pattern in both karyotypes. 2, 3, 4, they kind of lay them out from largest to smallest and then number them that way. But then the last pair is the X's and Y's. So yeah, the karyotype on the left has 2 X chromosomes and no Y chromosomes. So that's a karyotype for someone who is biologically female. And the one on the right has 1 X and 1 Y. So that's someone who's biologically male. So those first 22 pairs are referred to as autosomes. And I added that on this slide because I don't remember if we had that word. I think we had it somewhere else. But just to remind you of that, chromosomes 1 through 22 are called autosomes. So when we talk about inheritance of a trait, if we say that a trait is autosomal, that just means it's located somewhere on chromosomes 1 through 22, which all human beings have, and almost all human beings have 2 copies of each of those chromosomes. Those first 22 pairs are homologous, which means they carry information for the same traits. It doesn't necessarily mean they're identical. Obviously, you could have gotten a different gene you could have gotten the dominant gene from one parent and the recessive from another parent, but they carry information for the same traits at the same loci, at the same positions along those chromosomes. The 23rd pair, that last pair, are the sex chromosomes. They're sometimes also referred to as allosomes. Some people don't like the term sex chromosomes, even though that's kind of the most common name, because it implies that all of the genes on them have something to do with the development of sex characteristics, which isn't true. There are genes on these chromosomes that code for other characteristics as well. Males have an XY, and those two chromosomes are not homologous. The Y chromosome is not just an X chromosome that got cut off short. It does not have the same genetic information. There are genes on the Y chromosome that are not found on the X chromosome and vice versa. Okay, so they're completely different. So males have 22 homologous pairs of chromosomes and one pair that is not homologous. Females are XX, and those are homologous chromosomes, so they have 2 copies of each of those genes. So when you're talking about genes that are found on the X or the Y chromosome, obviously the inheritance of those genes is going to be a little bit different because some people get 2 X's and some people get an X and a Y. And so we have to kind of take that into account when we're doing things like Punnett squares and problems and things with sex-linked traits. So Y-linked genes, that means genes that are located on the Y chromosome somewhere. There aren't very many of them. There are some, but you saw it's a much shorter chromosome, so it just doesn't have as many genes. Many of them do affect the development of male reproductive organs on the Y chromosome. You will never see a Punnett square or inheritance type question about a Y-linked gene because the inheritance of the Y chromosome is always really obvious. If a dad has it, he will give it to all of his sons and none of his daughters. So any genes that are on the Y chromosome will pass to every male in the family and none of the females, right? So you never see probability problems or anything like that with a Y-linked trait. If we're talking about people with a normal number of chromosomes, then yes, because the only way that a boy child and a male child can get their Y chromosome is from their dad. There are actually quite a few variations in the sex chromosomes, which we're going to get to. You're going to learn about that a little more tomorrow, and we'll talk about it again on Monday. But there are people who are XXY and XYY, and there tends to be more variation in this pair of chromosomes than in the rest. And like I said, we'll talk about why that is on Monday. But if we're just talking people who have the usual 46 chromosomes, then yes, a male has to get that from their father because their mother wouldn't have one. Okay, X-linked genes are the ones located on the X chromosome. For a recessive X-linked gene, females need to have 2 copies to show the phenotype, but males need only 1. So sometimes one of the things that you will see with an X-linked gene is that phenotype, that trait, will show up more frequently in males, because if males inherit even just 1 gene that's recessive that causes whatever condition this is, they will show that trait. It's not possible for a male to be heterozygous for an X-linked trait because they only have 1 X. Whatever gene is on that X chromosome, that's the gene that's going to be expressed. Does that kind of make sense? And that's what I just said. Males can't be heterozygous. There are quite a few traits that are like this. Duchenne muscular dystrophy and hemophilia are both X-linked recessive disorders. That tend to be more common in males than females for this reason, because males only need 1 copy and females need 2 in order to show that disease or to have that disease. Okay, one practice problem with sex-linked traits for you. A human female carrier is heterozygous for a recessive sex-linked trait causing red-green color blindness, and she marries a colorblind male. What proportion of their male progeny, that's not a word I use very often, offspring will have red-green color blindness? Take a minute, write it out, talk about it, see if you can figure that one out. let's work through this one. As you wrote this out, did you start by writing X's and Y's? That's where you should start if it's a sex-linked trait, because that will help solve the discussion that you guys were just having over here about how many genes each person gets. Then you add the gene for the trait. So usually the notation used for a sex-linked trait is that you write the gene, the capital or lowercase letter that represents the allele that they have, as a superscript, like an exponent, next to the X or Y to show which gene it's attached to. This is an X-linked trait, which it has to be because the female is a carrier, so it has to be an X-linked trait, not a Y-linked trait. And it says the female is heterozygous, so she's got to have a dominant allele on one of her X chromosomes and a recessive allele on the other one. She marries a man who is colorblind, and it tells you that is a recessive sex-linked trait. So on his X chromosome, he has to have a recessive allele. A gene is only found on the X chromosome or the Y chromosome, not both. So we don't put anything next to the male's Y chromosome. You have to put the Y there because he has a 50-50 chance of passing that on to his kids. Then you can put this into a Punnett square with the X's and Y's and B's and everything. So the female will either pass on the first X with the dominant allele or the second with the recessive allele. And the male will either pass on his X chromosome or the Y chromosome. So then you can go through and fill this all in. The one other tricky thing about sex-linked traits is make sure you read the question really carefully. This asks what proportion of the male offspring will have red-green color blindness, which means when we look at this Punnett square, we're only looking at the males because it's just asking what percentage, portion, fraction of the male offspring will be colorblind. So out of just those two that are XY, what percentage or fraction will be colorblind?
25-50%.
Out of just the males. That's what I mean. That's why I have to read them carefully. 25% of all their kids will be males who are colorblind. 50% of the males, which is what the question said, will be colorblind.
Will it say, like, just reading it too, I guess. When it's like all of them, like what will it say?
Of the offspring, it would probably say something like, what proportion of the offspring will be males who are colorblind? So that would mean out of all of them, what percentage would be both male and colorblind? Does that make sense?
Yeah, that's good.
So yeah, read these questions carefully. The wording matters in how they ask those questions. There are one or two sex-linked trait problems kind of mixed in on some of the practice problem sets, and the pedigree problems do ask you if the traits that are shown in those pedigrees could be X-linked or Y-linked, so you can kind of think through it a little bit there as well. Last couple of things to wrap this up. As people started to learn about chromosomes and the fact that chromosomes carry the genes that determine our traits, and as they started to watch how chromosomes move during meiosis and how these get divided, they also started to realize that chromosomes can be the cause of some problems, some genetically rooted problems as well, because there are a lot of things that can happen to alter chromosome structure. You have examples of several of them here. And these can happen at several different points. They can happen during the process of crossing over at the beginning of meiosis. They can happen when the chromosomes separate. Sometimes they can just happen during the course of a cell's life as the genetic material kind of twists up and moves around within a cell. You can have a deletion. That's the first one. You can see the gene over here has A, B, C, D, E, F. This one, the B gene, got deleted completely, so that little segment of the chromosome was lost. It could have been damaged, it could have just been mutated, or it could have actually been cut out and lost completely. Genes can also be duplicated. So here you had one B, and here you have two. And like all types of mistakes and mutations, depending on what these genes code for, this may or may not have a detrimental effect or any effect on the individual, but often it can. You can get an inversion, which is when a chromosome kind of gets twisted up with itself and the ends switch. So you can see in this one, the location of A and F actually swapped. That's an inversion. Or a translocation. You have two homologous chromosomes in each cell. There are two copies of each chromosome. If two different chromosomes can kind of get mixed up, they can also swap genetic material. So let's see, ABC, so these, GHI swapped with these. Here's one really interesting thing about translocation that I just learned about fairly recently. First of all, I don't know how common translocations are, but I think they're actually more common than we would expect. I have a cousin who is perfectly healthy and normal. She has all of her chromosomes and all of her genes, but she has a couple of translocations within her chromosomes, like part of chromosome 5 is over here on chromosome 7, and part of 12 is up on 3. That happened a couple times. I don't know why or how or anything. She didn't know about it until she tried to have kids. Because then when these chromosomes separate to end up with an egg cell that has one copy of every gene, when the genes are all mixed up in the wrong places, the chances are really low. So she had a really, really hard time. They actually ended up adopting because they couldn't get pregnant. She never had any egg cells that had the right genetic components because she had all these translocations, but she herself was perfectly fine. And she has all of her DNA. It's just not in the normal spots. And I have a sister-in-law who has something similar. She just doesn't have it quite as frequently. I think she just has one that switched, and her mom had it as well. It's like a family thing. So those are fairly common. Interesting, huh? Some of these so here are just some examples of some conditions that result from some of these alterations of chromosome structure. Cri-du-chat syndrome, that's French for cry of the cat, because I think those babies, when they cry, sound like cats, so that's why they called it that. You can look up more about those if you're interested. The other thing, last slide or almost last slide for today, because this leads into what we're going to talk about after the quiz tomorrow. There can also be cells that have an abnormal chromosome number. That's called an aneuploidy. It results from non-disjunction. So during anaphase, that's when the chromatids are supposed to separate from each other and go to opposite ends of the cell. If they don't, you might end up with two chromatids going one way and none going the other. So you end up with one gamete that has an extra chromosome and one gamete that's missing a chromosome. Monosomy, mono means one. So it means instead of having two copies, there's just one. So they're missing a chromosome. Trisomy means three. So instead of just two copies like they're supposed to have, they have three. Down syndrome is a trisomy. It's trisomy 21, I think. And I think that's on the next slide as an example. It is also possible, not in humans. Humans would not develop as polyploid embryos. But apparently it's pretty common in plants. I don't know why they do this when we don't. To have entire extra sets of chromosomes. Bananas, as we have bred them over the years, are triploid. They have three copies of every chromosome instead of two. It might be part of it. I'm sure it happened in that process. This is actually also something that frequently results in the creation of new species of plants. Is this like doubling of the entire set of chromosomes? Strawberries are my favorite. They're octoploid. I just like that one. All right, human aneuploidies. There aren't very many that we can tolerate. There are some with the X's and Y's. Like I said, we're going to talk more about those. But in the rest of the chromosomes, the only aneuploidy where people can really live a somewhat normal length lifespan is Down syndrome, which is a trisomy of chromosome 21. So they have three copies of that chromosome. There are some others. There's like a trisomy 15, and I think there's a trisomy 18 maybe. And those children sometimes can be born, but they often don't live very long. They don't live more than a year or two because they have very, very severe side effects. So we're going to talk more about aneuploidies after the quiz tomorrow. So that's it for today. Kylie and Ashley, you are welcome to sign off, and we will see you again tomorrow. Interesting stuff, huh?